This video presents the simultaneous amplification of DNA samples from different patients. 2 microliters of each DNA sample is placed on 0.2 milliliter PCR conical tubes, numbered beforehand, correspondingly, and Eppendorf pipettes are used with individual tips for each sample to avoid contamination. Then, in another tube, a mixture is prepared as follows. A pair of short primers in large excess is added to a definite amount of distilled water. The primers are 2 synthetic specific oligonucleotides that bracket a target segment. One primer can hybridize to the 3' end of the target on one strand. The other primer hybridizes to the 3' end on the complementary target strand. Then the 4-deoxyribonucleoside triphosphates are added for synthesis of new chains. Next, Taq polymerase is added. Taq polymerase is a temperature-resistant DNA polymerase from a thermophilic bacterium from hot springs, Thermus aquaticus. This enzyme works well at over 70 degrees Celsius and is not denatured by temperature of 95 degrees Celsius. Magnesium chloride is added due to magnesium ion is cofactor of DNA polymerase. Buffer is added as well. The solution is mixed well by the pipette, taking care to avoid air bubbles. A definite amount of the mixture is added to the DNA samples. The tubes are closed carefully and the lids are numbered correspondingly. All tubes are placed for a short time off briefly in a centrifuge to mix well the components and all drops to move to the bottom of the tubes. The tubes are placed in the PCR thermal cycler that is closed well and the temperature regime is chosen to carry out the PCR. The program includes 35 cycles and the temperature is changed from 94, 58 to 72 degrees Celsius for a definite time in each cycle. The first step of the cycle is strand separation or denaturation or melting by heating the parent DNA molecule to 94 degrees Celsius. The second step is hybridization of primers. The solution is then abruptly cooled to 58 degrees Celsius to allow both primers to form base pairs with the DNA strands. Parent DNA duplexes do not form because primers are in large excess. Usually primers are 20 to 30 nucleotides long. The target sequence is between the ends of the primers. The third step is elongation of the primers. The solution is then heated to 72 degrees Celsius, the optimal temperature for Taq DNA polymerase. The enzyme elongates both primers in the 5' to the 3' direction. DNA synthesis takes place on both strands but extends beyond the target sequence. The larger strands increase in number arithmetically, while the short strands representing the target sequence are amplified exponentially. The whole process takes 1 hour 48 minutes for 35 cycles. The system is cooled to 10 degrees Celsius at the end. The tubes with the amplified segment of DNA are taken out of the thermal cycler.